minimize the
Many of the steps we use in CAR-T processing and cell processing involve open connections, and these require biosafety cabinets in order to maintain sterility of our process. Biosafety cabinets typically are a Class A by European standard environment, and these biosafety cabinets, these Class A environments, have to be in a Class B room.
So tell me a little bit about why that environment, this particular hood that she's working in, is an aseptic environment.
Yes, aseptic environment. So it kind of goes back even into our clean room. We have to make sure we minimize the presence of particulate in the actual critical work environment. In this case, for cell therapies, everything we do with the cells will occur in the biosafety cabinet. The biosafety cabinet is designed with a laminar flow whereby high efficiency particulate air filters help are utilized to push air down onto the work surface, out to the back, and then to be recirculated out. And the idea is to minimize particulate.
Great.
Particulate becomes a vehicle, if you will, for microorganisms.
Sure.
Lower the particulate, the lower the probability of a microorganism landing in that critical area. So the idea, and this once again goes back to the importance and the respect I have for the technicians, she will stay in that biosafety cabinet for 3 to 4 hours until all of her grafts are complete. The idea is keeping that aseptic field so her hands stay in the environment and she's not coming out.
What we're looking at over here is called a sterile welder and a hand sealer. So these two pieces of equipment are our most basic and most often used pieces of equipment. So basically what they're used is to maintain our sterility and integrity of our process from end to end. So we have a few open steps in our process, which were done in the biosafety cabinet, and then we try to keep our process as closed as possible. And these two pieces of equipment help with that. If you have your drug substance and you need to take a sample for analytical for quality control, you can attach the sampling set directly to it. It maintains sterility, remove the sample without having to worry about compromising the sterility of your drug substance. This piece of equipment we use on a daily basis, routinely, dozens of times per process.
So Rachel, I would love to see how this tube welding works.
Sure. So the way this works is you take 2 pieces of tubing and you figure out what you want to connect. So
Okay.
let's say I want to take this bag and connect it to this lure.
Okay.
In that case, I would take this bag with the tubing, fold it in half so that on the top is the bag, which is what I want to connect.
Mm-hmm.
And the tubing is on the bottom, which I will want to discard later on.
Okay.
And I'll just place that into this insert. Make sure there's a little bit of tubing sticking out so that it's exposed to the actual spot where it will be welded.
Okay.
Clamp that shut so it's secure. Make sure it's nice and loose and not, trapped anywhere and pinched. And then same thing with this side. So I want to connect it to my lure, so I'll fold it in half and make sure my lure is on top.
And the waste tubing is on the bottom.
Correct.
Okay.
So make sure that's tucked in, clamped shut, nice and loose. And then I have a wafer here. So this is a blade that I'll place right here. What the instrument will actually do is heat this blade up and then move it forward and take these 2 pieces of tubing and have it close together so that the heated blade will cut through the tubing and then push the pieces of tubing together so that the connection will be sterilely welded together.
Ah.
So let's get started. So now once I open this back up, you can see that the tubing is pushed together. And just in case, usually I like to take a hemostat and clamp both sides. Just in case there is a problem with the weld and there's an opening, you want to make sure that whatever you have on the side is protected and so it doesn't introduce any contaminants from the air.
Okay, so that's like a safety clamp you're putting in there.
Correct.
Okay.
So take that, lift it right up so it's nice and connected.
All
And
right.
you'll actually see that the bottom pieces are also connected. So what you'll have to do is take the 2 top pieces together and the 2 bottom pieces together, pull them right apart, And usually I like to take a look at the waste pieces and make sure, if you pinch it, you can actually open up that weld, and now the path between the two pieces of tubing is open.
Okay.
So that looks successful for this one, and so now I can check the tubing that's on top. So push it, open up that line, and now you've got your bag connected to a lure sterilely.
Fantastic.
So there are different kinds of welders. The welder that we're going to use today is a little bit more bulkier. But most welders tend to work very similarly. This particular welder, all welders will have either an adapter which can connect different size tubes.
Mm-hmm.
In this case, most of the kits that we use tend to use these tubes, which are a 1/4 inch by 1/8 inch sometimes. So this adapter over here is perfect for that. If you do work with tubes that have wider diameters, then you would either get a completely different welder that can handle that, or you might, in some cases, you might just need this particular adapter. So in this case, it's showing me on this side, this is my bag. This is the bigger end that I want. So I'll connect that, I'll put that in like that. And you can go ahead and I can bring this closer to you.
Okay.
Yep.
And does this qualify as a wet weld because there's liquid in one of the lines?
Yes.
Okay.
And this machine can be used for dry welding or wet welding. If the things that might go wrong, you could sometimes pinch the line. So if you do not add it incorrectly or do not connect it correctly, you could pinch the line in that sense. That can, the pinch itself can open up the line, expose it to the outside air, so it will not necessarily be being sterile. And that's on both sides.
Mm-hmm.
Or occasionally what could happen with most welders is, if there's not a little bit of slack, when the weld, after the blade cuts through the plastic at a high heat to melt the two tubes together, if there's no loose, if it's too tight, if there's too much taut or pressure on the line itself, what will happen is as it's trying to pull the two together, you might get this pullback, and so the seal doesn't happen properly.
Okay.
That's a very common error that can happen. That's why it's really important to make sure you have some sort of slack on the line.
Okay.
Here's a nice summary as well of what happens. And so then all I do is I press start. The light goes green. And another thing that could happen is you don't want to open this until the machine will beep.
Okay.
And that will tell you that it's done. As is what happened there. If you stopped it at any point, again, you would have exposed the lines without sealing it. Now it's ready to go. So you go ahead and open it. So you'll take this.
And in this particular model, the waste is left behind. You don't have to break off the waste tubing.
Correct. On this end, we've now performed the seal. And to make sure that it's successful, we'll add the hemostats on either side. And then we are gonna open the seal to make sure that it's still sterile.
Okay.
If for any reason it wasn't somehow you pinched it, or you feel like it wasn't sealed properly, you can then use the sealer again to seal on the outside of where you put the hemostats, and that way the system should still be sterile.
Okay.
On either side. So what I'm doing now is just checking that the seal is clear and it looks good. The nice thing about having a liquid in there, is if there's a leak at all, you will detect it immediately.
And you also know that you have it open because you can see the liquid going from one side to another without leaking out.
Correct.
Okay.
Once you are happy with that, you can take off the hemostats. Or whenever we want to remove anything from a tubing system, we use a sealer.
Okay.
And literally what the sealer does is it just heats up. There's a plate inside there. It will pinch the tubes together and generate a heat that will cauterize that particular thing, and then you can pinch it off.
Okay. So it's similar to the tube welder, but you're not connecting two different tubes. You're just sealing one off.
Correct.
Got it.
So I'll go ahead and do this. So you have this seal like that,
Okay.
and you can just pinch it off. And so now it's sealed and the system should remain closed.
So Chesney, you made this look really easy, so it's clearly sealed and it broke off cleanly. But what are the ways that tube sealing could go wrong? Because there's a big consequence to it going wrong. You just opened up the system to non-sterile conditions.
Correct. So one of the ways that this could go wrong is if you stop the seal prior to the sealer telling you that the seal has completed.
Okay.
So, for example, you can put in the seal. As soon as I push this button, the light will switch on. Mm-hmm. If I just remove my hand before the seal has completed, that's one of the ways that it could fail.
Okay.
There are occasions where you could push the button and not within your power, but the sealer itself could just decide to abort. Uh-huh. And if you're not keeping an eye out on that, that could cause a failure as well. Again, most of the time, it's really successful. So another problem that could happen is if you do not add the tubing incorrectly. And so what will happen is the plates within the sealer might only seal half of it. So maybe I can demonstrate here. So that's one problem where you could go in, you might not have paid attention to the whole system, and you just open it up and you could just create a tear. Okay. So it's always good to make sure that the tube is in correctly and that you follow the prompt that it's now done, and then go ahead and you can open that seal.
Oh, thank you very much. Even that takes a little practice. All right.
There we go.
Great.
Thank you.
It is a relatively simple piece of equipment, so obviously it is calibrated, and basically the calibration just is a sterility integrity test. So that connection, that sterile weld that we just created, essentially our equipment group would perform that function a number of times and make sure those connections are consistently true, they're consistently well joined and maintain sterility.
We're looking here at some people who are gowned up. Can you tell me a little bit more about what they're doing? Are they wearing this gear in particular because they need to be protecting the product that they're working on or themselves? Why are they gowned up?
Yes. So in this environment, we have basically a grade B ISO 7 clean room. So we have to control our environment to produce our biologic, in this case, our epicell grafts. The issue is with a biologic or a cell therapy product, we cannot terminally sterilize the final product. Thus, we need to use environmental controls to establish a facility that minimizes the possibility and potential for microbial contamination. Then we have a dedicated biosafety cabinet to once again further produce as clean an area as possible for the production of our cell product. In this example here, we have our two senior technicians, Cornelia and Donald, actually assembling epicell skin grafts.
The folks that we're looking at here who are working in these hoods, did they have to go through any special training, or how did they come to be doing this type of work?
There's a very involved training program to bring people into a clean room environment. Mm-hmm. We begin with a full gowning qualification, gowning program to teach them how to gown in an aseptic fashion to minimize the potential of them introducing microorganisms into the clean room environment. They then go through numerous training steps on how to work aseptically in a biosafety cabinet, and then ultimately will be qualified on working with the actual skin cells.
So let me ask you one more question about the environment that we're looking at here. So this is a sterile environment, everyone's gowned. How are you measuring or monitoring the room to assure that things are sterile and that the products are sterile at the end of the process?
We rely on our quality control group to help oversee control of our facility by their environmental monitoring. They will measure the air for viable and non-viable particulates. We will use auger plates, settling plates to assess the potential for viable particulate in the biosafety cabinet environment. Mm-hmm. We will also sample and touch the technicians, their fingertips, their forehand, their hands, their wrists, etc., to identify are they potentially bringing in microbes in, or microorganisms into our actual environment.
The activation step is where we tell the cells, by using some antigens, that they need to start working again. The idea is to promote growth and proliferation of the cells, because we want the cells to multiply.
And then we activate these cells with CD3 and CD28 antibodies and give them IL-2 to help them grow. And this is an environment that encourages T cells to grow and doesn't really support other types of cells. So after a couple days, most of the other cells have died off, and you have a very T cell rich population.
Parameters that are considered on this step is the concentration of cells. How many cells are available for the manufacturing process, and how healthy are those cells.